Okay, I have 12 o'clock, so let's go ahead and get started. This is Microbiology, Bio 120. I'm Dr. Van Laar. I'll be your microbiology instructor for the rest of the semester. Microbiology is one of my favorite classes to teach, one of my top 2. So this is going to be fun for me. I hope it's fun for you. If not, I'm sorry. But you still have to be here anyway because you're in class, so good luck. But I like it. I think it's really fun. I teach it fairly often. Actually, I taught it just last fall, so I'm teaching it 2 semesters in a row because I like it just that much. I am a microbiologist by training. I'm a medical microbiologist. I work on bacteria that cause severe human infection. I work on antibiotic resistance, which is what you'll be studying in the lab. So I like bacteria. We're mostly going to talk about bacteria, but we will talk about viruses and fungi and a little bit about some other types of parasites as well. But again, mostly microbes. This information is going to be related to things you hear in genetics, to things you hear in cell bio. We'll talk a little bit about ecology and evolution. So microbiology is one of those fields that can kind of tie everything together. Hopefully you're already all enrolled in a lab section. If you are not enrolled in a lab section, unfortunately this class is full. We have 5 lab sections, max 20 people per lab section. Each is maxed out. So if you are on the wait list, you have to hope that somebody drops. That almost never happens because this class, as you know, fills up pretty quickly. If you don't show up to lab next week, I will drop you, and then somebody else can get your spot. Okay, so if you're on the wait list, you can hope that somebody doesn't show up to lab on Wednesday. Other than that, I can't do anything because I can't add extra people into the labs. You can all fit in here and that's fine, but the labs only hold 20 each. Okay, so any questions about that? Okay, how many of you have read the syllabus? Most of you, yeah, good. When is the first exam? This Wednesday. Yes, this coming Wednesday. And I can always tell those of you who are like, yeah, I read the syllabus. And then we're like, okay, the exam is on Wednesday. You're like, oh my God. So you haven't read the syllabus. So you got to work on your facial expressions. Okay. So the first exam is on Wednesday. We have an exam every Wednesday. Okay. If you've read the syllabus, you already know this. This is not a surprise. Those of you who haven't read the syllabus, we have an exam every Wednesday. Your 2 lowest scores will be dropped. Therefore, there are no makeups. So I don't care if you were at a conference, I don't care if you were at an athletic event, I don't care if you were sick, I don't care if you were hungover, I don't care if you were just like, screw this class, I'm not going today, whatever. I don't care. Any reason you don't come to class is totally fine. 2 of your exam scores get dropped, regardless of even if they were 0. Okay, so I don't do makeups. We also have pop quizzes. It's a Discover E class. You have tablets or a computer or a cell phone. There are also no makeups for those because they do drop your lowest scores. So come to class, pay attention. I'll be here. I'm here, you should be here. I'll give you 50 minutes. You give me 50 minutes, we're good to go. People always ask me, what is the best way to study for this class? Your PowerPoint slides are online. You know what chapters I'm going to be covering. You should be reading these chapters in the book. If you have an older edition of the book, that's fine. I don't know the difference between the older editions of the book and the newest edition of the book. They only give me the newest edition of the book. So all I can tell you is this is what I'm going to be teaching you. If it's not in your older edition of the textbook, you'll have to read it online somehow. But you should be looking at this before you come to class. If this is the first time you are seeing this slide, you are already behind. We recommend in every one of your science classes that you spend, for every hour in class, 2 to 3 hours a week studying. This is a 4-unit class. You spend 3 hours a week in lecture, so you should be spending at least 6 hours a week studying. If I spend that much time preparing for this class, I have a PhD in this. Right? I spend that much time on this class, you should be spending at least that much time on this class. So when you come to my office and you ask me, why am I not doing well? I'll ask you your study habits. And when you tell me, well, I study like an hour a week, there's where we're going to start. If you're studying 6 hours a week and you still have problems, we'll reevaluate how you're studying. You can always come to my office hours. They're on the syllabus. So you can find that on the syllabus, which you may or may not have read. That's fine. It's on Canvas. So you can always come to my office hours. You can always send me an email. Usually I'm pretty good about responding to emails. except for on weekends, because as much as I like to pretend I have a life, I'm still going to keep pretending I have a life, and I may not reply to emails on the weekend or on a holiday. Okay? So it's a lot of information, but you can do it. I almost never have a student fail this class. You really have to try. Like, you have to try to fail. Yeah. There's 100 students every semester. I think maybe I've taught this class 3 or 4 times and I've failed like 1 or 2 people. They just stopped coming to class. So, I mean, you don't come to class, you don't take an exam, you don't take the final, you're probably going to fail. But if you show up, you take your exams, you take the final, you won't fail. But the level of effort you put in is going to be reflected in your grade. Any questions on the syllabus, the course structure? You'll have a paper you have to write. It's in the syllabus. Today is January 17th. I don't remember exactly when the paper's due. It's sometime in April. It's January 17th. The paper's due in about 3 months, just FYI. So 3 months from now, please don't act surprised that I'll be expecting a paper. There's always one. Wait, our paper? Yeah, you have a paper. Okay. You'll have to pick a topic. The day the topic is due is in the syllabus. I will approve your topic. If you go to Canvas, there should be a link to a Google Sheet. If there's not, I'll put it up later today. Everybody writes about a different organism, so pick an organism. I don't care what organism it is. If it causes disease, I'm interested. It can be a virus, it can be a bacterium, it can be a fungus, it can be a parasite, whatever you like, whatever you want to write about. If you're interested in plants, pick a plant disease. If you're interested in animals, pick an animal disease. If you're interested in people, pick a people disease. Everybody picks people diseases, so pick fast. The flu goes quick. MRSA goes quick. Measles goes quick. Pick a topic quickly, and you'll be good to go. We'll also do journal article discussions, so I will require you to read primary literature. You will have to come to class prepared for that. Your peers will evaluate you on how well you came to class prepared. You won't know what they said. It's anonymous. I'll know what they said. So if you ever get stuck in a group with somebody who doesn't do their part, you tell me. I'll make sure they don't get points for it. Okay? So you can always call out people who don't do their part. So you read 4 primary literature articles for class. You'll read at least 4 to 5 for your paper. All of the instructions for the paper are already online. So get into Canvas, play with everything in there. If you haven't downloaded the syllabus, please do so, because that's how it's the contract for the whole class. I know it's in the contract. Hopefully you know it's in the contract, but we can't change the contract after today. So if you show up on Wednesday, I'm going to assume you agree to the contract. Okay? So what are microbes? Well, they're small things, generally. Most microbes we talk about, again, are bacteria. Most of the time when people think about microbiology, they think we're talking about bacteriology. There are other organisms that do fall under the classification of a microbe. Those can include fungi, which are eukaryotic. They can include some of our parasites, everything from unicellular protists all the way to, 30-foot-long tapeworms are technically considered microbes. And of course, viruses, which we'll talk a little bit more about viruses when we get to chapters 8 and 10. But the idea behind viruses that's kind of unique is that most scientists don't even call them living, but they do cause disease. When we talk about bacteria specifically, we have a number of different shapes that our bacteria can take. And when you're in the lab, you'll look at different bacteria under a microscope, and you'll see some of these shapes, but not all. So we have round or coccus. Some of our classic disease-causing organisms would include Staphylococcus aureus, right? Methicillin-resistant Staph aureus is your MRSA. Streptococcus pyogenes, or strep throat, right, are all going to be coccus-shaped. We have rod-shaped bacterium, so a little bit more elongated. A lot of these are some of our friends like E. coli, Salmonella, right, some of your gastrointestinal infections, but also things like Bacillus anthracis, which causes anthrax. Spirillum is kind of a comma or a curved bacterium. You'll sometimes see things like Vibrio cholera, which causes cholera, or Helicobacter pylori, which causes stomach ulcers. Spirochetes, they're my favorite. Sometimes we call them corkscrew-shaped. Everything from syphilis to Lyme disease is caused by a spirochete. And then we have some more unique shapes that we don't often see in medicine, but are really, really relevant to environmental bacteria, including some that can form stalks and hyphal structures, and even some that can form long filaments. These are going to be more common, especially with some of our photosynthetic microorganisms. A lot of times when people talk about photosynthesis, you think, well, plants, right? Plants are the most important thing because they produce so much oxygen. It's actually bacteria that produce the most oxygen. Bacteria do everything better. They do everything we can do, and they do it better. So they do photosynthesis. They do more of it. They are the primary producers, actually. So we'll have different shapes. You will see some of those under a microscope. Bacteria are very small. For the most part, most bacteria are going to be smaller than most eukaryotic cells. And so our size range is shown here. I'm not necessarily going to ask you to remember in micrometers how big are different bacteria. Right? Doesn't matter. In general, most bacteria and most archaea, our prokaryotes, are smaller than eukaryotes. Eukaryotes have a nucleus. Eukaryotes have all these other organelles. Eukaryotes kind of have to be bigger because there's a whole bunch of stuff crammed into them. Prokaryotes don't have organelles. Prokaryotes don't have a nucleus. Prokaryotes can be smaller. Most fall in what we call kind of an average range of size. There's too many lights in here. Let's see if I can adjust that. But we do have some prokaryotes that can be very, very large. Again, I'm not going to ask you to remember, oh, well, which one specifically is the really large one? Like, not important. Okay? If at any point you really wanted to show off to somebody and tell them what the largest prokaryote is, you're more than welcome to look it up in the textbook. But again, most eukaryotic cells are much larger. We do have small eukaryotic cells. Sperm cells are very small. Red blood cells are very small. Red blood cells don't have a nucleus, at least in humans and other mammals, so they're small. Okay, but again, for the most part, general rule, prokaryotes, which include bacteria and archaea, are going to be smaller than all of our eukaryotes. And again, here's a little size comparison of our largest bacteria all the way down to our smallest. So some of our smallest bacteria are found in the ocean or are uncultured, but there are some very small bacteria that can even cause human disease. The one down there on the bottom is Mycoplasma pneumoniae. It causes a disease that we sometimes call walking pneumonia. And as we progress through the semester, we're going to talk a lot about different features that make microorganisms microorganisms. We'll talk about things like the way they organize their cell membranes. And we'll talk about things like their cell walls. And I'll do my best to tell you why those things are important. Right? Not just, well, you need to know this because it is microbiology, but you need to know this because it is relevant to you for these reasons. For example, if we talk about Mycoplasma pneumoniae, this is a really weird little bacterium. It does not have a cell wall. Almost all other bacteria have a cell wall. So why is that important? Who cares? Well, if this is an infection you have, or if this is an infection you are trying to treat, there is an entire class of drugs that you can't even use. Things like penicillin, which inhibit the cell wall. You don't have a cell wall, you're resistant to penicillin. So you have to find a whole new class of antibiotics. So it's really important to understand the basic biology of these organisms, even if you do want to be a doctor. So basic biology can be really, really relevant. And if you don't want to be a doctor, it's still important to understand basic biology, but you probably find it a little bit more interesting. Okay, here they're showing, this is your large prokaryote. This is a paramecium, which is a eukaryote. So you can see that there are some prokaryotes that are larger than eukaryotes. Okay, What does that mean then in terms of how these organisms can function? So why are prokaryotes smaller? Well, there's less stuff in them, so they don't need to be larger. But what other benefits does that have? When you look at something that is small, it has what we call a high surface area to volume ratio. And we can do some math with like spheres in a little bit. We'll see that on one of the next slides. But basically the idea is when you are very small, your volume is also kind of small, but your surface area might be a little bit bigger. And when you have that high area of surface, the really nice thing is that all of the processes that have to happen on the surface, you have a whole big area in which to do those. So you want to bring in nutrients. You have this huge area to bring in nutrients for a tiny little cell. You want to get rid of waste. You have a huge area to get rid of waste for a tiny little cell. But the bigger you get, the more volume you have, that ratio goes down. And that means you're trying to do nutrient exchange or gas exchange or get rid of waste products for a big, big, big cell in a tiny little area. And so you often cannot do things as efficiently. So smaller cells can have a greater nutrient and waste exchange. And that means they often grow faster. So again, a kind of general rule of thumb, prokaryotes are smaller than eukaryotes. Another general rule of thumb, prokaryotes grow faster than eukaryotes. And when we talk about single-celled organisms and we say the word growth, we usually don't mean getting bigger in size. We mean getting bigger in number. Right? When we talk about people and we say people grow, we mean you either grow up or you grow out, right? You get bigger. You take up more space. But again, when we talk about bacterial growth or archaeal growth or even single-celled eukaryotic growth, we're talking about replication. So I'm saying that bacteria, archaea, replicate faster than a eukaryote. Some bacteria that cause human disease, like how many of you have heard of C. diff in hospital settings? Right? Clostridium difficile, the organism, can replicate in about 7 or 8 minutes. So you go from 1 to 2, 2 to 4, right? 4 to 8, every 7 or 8 minutes. A U cell takes like 24 hours maybe to go from 1 to 2. And the Clostridium difficile is kind of an extreme example. How about E. coli? You guys know about E. coli, right? In about 20 minutes, kind of slow. Staph aureus, 40 minutes. Kind of slow. Faster than you, though. Some of the slowest bacteria, like Mycobacterium tuberculosis, which causes tuberculosis, or Mycobacterium leprae that causes leprosy, they're very slow. About 24 hours for them to replicate. So they're about the same speed as you. But again, most prokaryotes are faster than eukaryotes. And that will provide them extreme advantages. Because if we think about how do we adapt to a threat, usually we have mutations, right, that will manifest themselves as different phenotypes that will eventually allow you to adapt or evolve. If you make a whole new organism every 8 minutes, you evolve pretty quickly. How long does it take humans to make a new organism? Like 20, 30 years before humans might make a new organism? If you decide to make one at all. Right? So human evolution is very, very slow. We can't see human evolution. If you have children or if you're planning to have children, when you look at your children, you won't know genetically how they're different from you. What mutations do they have that may allow them to adapt? When you expose bacteria to antibiotics, you can literally see them become resistant in about a week and a half. So I'll show you a cool video. Cool and scary all at the same time, which is a lot of microbiology, I think. So being able to grow faster has an advantage. And they can get to huge numbers. I do clean this, so I'm going to use this as an example. It is clean. It is just water. But let's say I put something in here that bacteria like. Even if it was like milk or juice or something, it's just water. This is about 3/4 of a liter. If I put one cell of E. coli in here right now, and I came to it in 24 hours, I would have probably 1 trillion cells per milliliter. And this is 750 milliliters, so 750 trillion bacterial cells in here overnight. What are the chances that at least one of them will be able to tolerate whatever kind of threat I give it? Pretty good. 750 trillion, right? The idea that a huge population like this will have mutations that will allow them to survive threats is why bacteria are going to eventually take over the world. We simply cannot evolve fast enough. Okay. This is the largest public health concern, according to the World Health Organization and the CDC, is the evolution of antibiotic resistance. So antibiotic resistance is what you'll be looking at in the lab. The World Health Organization estimates that something along the lines of 10 million people per year will die of antibiotic-resistant infections by the year 2050. That's more people than are killed by cancer. It will put something along the lines of 30 million people additionally into poverty because they can no longer work, they lose their breadwinner, they're bankrupt by medical bills. Those will be all Americans, right? Bankrupt by their medical bills. In other countries, they may lose their primary breadwinner, they may not be able to work. Here we get bankrupt by medical bills. We literally have situations where scientists come up with a new antibiotic, they put it on the market, and in less than 1 year, you have antibiotic-resistant bacteria because they grow so fast. They always can outcompete us. So if you want job security, be a microbiologist. So long story short, I know, too late. Mutations lead to faster evolution. Every time, if you have taken genetics, you know this, every time you copy your DNA, there are going to be errors. It's pretty good. DNA replication is pretty good. But there is an error rate. It may be a very low error rate, but it does exist. Those errors are mutations, and mutations drive evolution. When you replicate every 15 minutes and you go from 1 to 750 trillion, it's any wonder that we've even made it this far at all. Eukaryotic cells adapt slower, especially us. Some of these unicellular eukaryotes, though, they do pretty well. There was a report that came out by the CDC, a few months ago. It was in 2019. It was last year already, so it's really out of date. Well, with microbiology, it really is. Some of the fastest growing health concerns are multi-drug resistant fungi. So you get a fungal infection that'll kill you instead of a bacterial infection that'll kill you. So even though fungi are eukaryotes, some of these fungi still grow faster than we do. They accumulate mutations that allow them to be drug resistant. Okay. Here is a diagram showing you a surface area to volume ratio. If we were looking at 2 cells, let's say, 2 spherical cells, or coccus-shaped cells, the first one has a radius of 1, the second one has a radius of 2. We can calculate the surface area using this formula right here. I'm sorry, a little bit of geometry, that's okay. I won't ask you to remember the formula, but just so you know, it's here. Surface area, 12.6. Volume, 4/3 pi r cubed. Volume, 4.2. So your surface area to volume ratio for this small cell is 3. You increase the radius by 1. Basically double the radius, which, I mean, it really increases the surface area quite a bit, but it also increases the volume even more. And so your surface area to volume ratio goes down to 1.5. because you're looking at a square and a cube. So of course, if you cube something, right, it gets bigger faster. What they're trying to show you here again is just this idea that the larger a cell is, the larger or the smaller the surface area to volume ratio is. So at some point you do max out where you can no longer have a cell of X size because you simply won't be able to exchange waste products, to get nutrients, to transport things like oxygen fast enough to promote cell growth. We also can reach too small of a cell because at some point you won't have enough room for the DNA and ribosomes. But we can get pretty small. So our lower limits of cell size, based on scientists' best hypotheses, the lower limits of cell size are probably going to be around 0.15 microns in diameter. If you get any smaller than that, you're unlikely to be able to have enough space to hold your DNA and to hold your ribosomes. Okay? So that's why. You just don't have enough space. Picture a tiny home. At some point, the home is too tiny if you can't fit in it. Or think of the bare minimum things you need in your home. Most people would like a restroom, maybe a small place to store your food, and a place to sleep. So how small can you get to have all of that stuff still in your house? So you do reach a lower limit. At some point, you might make a house so small that you can't fit a stovetop and a toilet. And that's not a trade-off I'm willing to make. I want both of those things in my house. I'm a little bit more demanding than that, but that's fine. Open oceans is where we tend to find some of the smallest microorganisms, and they call them ultra microbacteria. The really, really tiny ones. And again, as we talk more about microbial ecology and microbial evolution, we'll talk more about organisms that we find in those types of environments. So how did they get to be so small? Well, usually they streamline their genomes. And they might be missing functions that sometimes we would consider to be essential. So if you're missing something that you consider to be essential, how then do you survive? There are options. One option for some very, very small bacteria that have really, really streamlined their genomes quite a bit is to become a parasite. It's a life strategy. I can't do X, Y, and Z, but you can, so I'm going to be your infection. It works. It's a whole life strategy to be a parasite. Other times, maybe they're not so selfish, and they say, well, I can't do X, Y, and Z. You can't do A, B, and C. Why don't we be friends? And they can form symbiotic relationships instead. I guess psychologically as people, we probably would prefer that one, though I think we probably all know a few parasites too. So those are strategies. You can live in tandem with another organism where you provide something, it provides something, and you have this nice relationship. Or you can be a parasite, where you do not provide anything and you just take. They're both valid life strategies. There are literally thousands of organisms that have adapted both. Okay. So these are common strategies for some of these really small organisms to survive. Okay, so what makes up a cell? We've been talking about cells, bacterial cells, there's eukaryotic cells. What are some of the features that make up a cell? Well, the first is the membrane. Cells have a membrane. When we talk about viruses, and I tell you viruses are not living, viruses are not cells. One of the reasons that viruses are not living and that we do not consider viruses to be cells is because a lot of them don't have a membrane. And when they do, it's different. It's not the same type of membrane. So this is a feature of cellular life. Cells have a membrane. And it surrounds something called the cytoplasm. Hopefully this is a review of like Bio 1A or Bio 1B. If you don't remember seeing this in Bio 1A and 1B, well, you're seeing it right now again, so hopefully that helps. The membrane separates the inside of the cell from the environment. And really its main function is to be this selectively permeable structure that allows in only what we want in and pushes out what we want out. So it's the barrier that protects the bad things from getting in and lets only the good things in. In bacteria, the bacterial membrane can look a lot like a eukaryotic cell membrane. It's a phospholipid bilayer. You've got your hydrophilic heads and your hydrophobic tails, just like we see in a eukaryotic membrane. It contains embedded proteins. Those proteins in the membrane can have a lot of different functions. They can help transport nutrients, they can help cells communicate, they can help cells move. Do you all remember what is the model called when you talk about a cell membrane?
Fluid mosaic.
Fluid mosaic, very good. Right, so you have this fluid mosaic model where the membrane is not static, it's not fixed. Proteins can move around, the lipids can move around, things can move around in that membrane, and that's absolutely critical to allow cellular function. When the membrane stops being fluid, the cell dies. This is one reason why when cells have evolved to grow best at moderate or hot temperatures and you freeze them, they die. They can no longer do nutrient exchange. They lose the fluidity of the membrane. But what we'll talk about is the idea that there are lots and lots of bacteria that do very, very well at cold temperatures, including some that cause human disease. So at some point, they had to have changes to their membrane to allow that fluidity to continue at cold temperatures. You lose the fluidity, you lose life. Again, it was what your phospholipid bilayer would look like in a cartoon. You have those hydrophilic heads, those hydrophobic fatty acid tails. And this is a transmission electron micrograph showing exactly what the membrane looks like in a cell. Okay, so we have a little animation. Ooh, let's see if the sound in here is as loud as I think it's probably going to be. Okay.
The cytoplasmic membrane of a prokaryotic cell is the main gateway for movement of molecules in and out of the cell. This membrane may also be called the cell membrane or plasma membrane. Cytoplasmic membranes in bacteria are comprised of a phospholipid bilayer. Each phospholipid contains a phosphate head located on the outer part of the bilayer, attached to a glycerol and 2-fatty acid, which face the core of the membrane. The phosphate is negatively charged and hydrophilic, while the tail portion of the phospholipids is hydrophobic. Thus, cytoplasmic membranes are very effective at blocking passage of many molecules, while other molecules are allowed to pass. Cytoplasmic membranes also contain 2 types of proteins. Integral proteins often span the entire bilayer and act as channels for transport of molecules that normally wouldn't be able to penetrate the bilayer. Peripheral proteins are loosely attached to the membrane on one side or the other. Some of these may be glycoproteins, which are proteins bound to a polysaccharide. These proteins may function as recognition sites, enzymes, or receptors.
Okay, so there's your review of the membrane and its function. Again, you can see a cartoon like this where you have integral membrane proteins. Sometimes we call them transmembrane proteins because they can span through the whole membrane. And you have your peripheral membrane proteins that might hang out on the inside of the cell or the outside of the cell. And there are lots of different functions. They can catalyze reactions, help with transport, allow for cell signaling, allow for cell binding. So it's absolutely critical that we have this membrane, that it has its fluidity, so that we are able to maintain life as a cell. So we have this other group of prokaryotes. If you remember your 3-domain structure, right? We have 3 domains of life: bacteria, eukarya, and archaea. And sometimes people are like, oh, well, you know, biology is about like plants and animals. And then there's also the microbes. I get really offended. I'm easily offended, so it's okay. We have 2 full domains of life that don't even have a nucleus. And everybody's so concerned about all the stuff that has a nucleus. It's like 1/3 of the domains. And actually, scientists are now considering going to a 2-domain system. 1 domain of bacteria, 1 domain that contains archaea and eukaryotes, because eukaryotes are simply an offshoot of the archaea. So when you start thinking you're high and mighty, you're not. You're only a subdivision of archaea. But archaea have weird cell membranes. Sometimes people call them extremophiles. You can find these in hot springs, deep sea thermal vents. So we usually think of archaea as being organisms that inhabit these niches that we previously thought were uninhabitable. As far as we are aware, there are not archaea that cause human disease. Does that mean they don't associate with us? Absolutely not. The microbiome, the collection of microbes in the body, has been shown to contain archaea, especially in animals that do fermentation, like cows. They have a lot of archaea in their gut. But as far as we know, they don't cause disease. So they have a modified membrane. I'm not going to ask you all the details. You can look at the slide. I also record all of the lectures, so you can go back and listen to my voice. Some people, they're like, oh, I listened to it on the way home. I'm like, don't do that. Not if you're driving. Fall asleep. If you can't fall asleep, though, you can play this, and it's like a meditation, okay? I used to read my OChem textbook when I couldn't sleep. Don't tell any of the OChem professors. Okay, so when I couldn't sleep, I read OChem. Maybe you'll listen to me talk instead. So they replace their fatty acids with a slightly different compound. They have different major lipids. Again, I'm not going to ask you all of these details. As we talk more about how these organisms have adapted to their different environments, we'll get into a couple more specific differences that you'll need to focus on. But one of the other major modifications that some archaea have, not all, some archaea, instead of having a phospholipid bilayer, have a phospholipid monolayer. One of the advantages of this is that for those microbes that live at really, 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 really hot temperatures, this bilayer can separate. But if you have a monolayer, it can't separate. So for the archaea that have a lipid monolayer instead of a lipid bilayer, they're very well adapted to high temperatures. But we don't know a lot about archaea yet. Archaea have historically been very difficult to culture in the lab, so learning about their biology has been tricky. A lot of what we know about archaea is because of their DNA. We find their DNA places, we sequence their DNA, and we make predictions about what archaea might do. But as we have advances in the laboratory and we are better able to culture some of these organisms, we will learn a lot more about their biology. So again, functions of the cytoplasmic membrane, especially in our prokaryotes, the permeability barrier, a home for proteins that help us interact with our environment, and also energy conservation and generation. Where do eukaryotic cells do see energy conservation and generation? The mitochondria. Prokaryotes don't have mitochondria. So they do all their ATP generation in the cytoplasmic membrane, in the cell membrane. Okay, so eukaryotes, because they have a mitochondrion, at least one, depending on the cell, they may have thousands. Because they have mitochondrion, they don't do ATP synthesis at the membrane. But prokaryotic cells do. And so when we talk about the permeability of the membrane, we can also talk about how fast things can and do enter the cell. Through simple diffusion, it's usually pretty slow, because simple diffusion, you're really kind of relying on this random collision of a molecule with the membrane. You're hoping that the concentration differential works in your favor, and eventually you'll get things to kind of trickle in. When you have a protein in the membrane that is a transporter, you can then begin to increase the rate of materials entering the cell. So a low affinity transporter will grab things from the external environment and bring them in. But it's kind of like, Butterfingers, right? You throw a ball to somebody and they catch it like half the time. You hope that guy's not on your fantasy football team. Yeah, I hope he's not on the 49ers, right? We got to have good. Sorry, I'm a 49ers fan. If you're a Packers fan, well, I hate you, and we'll see what happens next week. Right? You hope they don't have Butterfingers. If they have Butterfingers, they're not going to score a touchdown very often, right? Or if you like baseball, if the guy's dropping a ball in the outfield, half the time it's thrown or hit to him, you're going to be really ticked off. You're going to want that guy off your team. So only some of the time will things get in. But if you have a high affinity transporter, is where you hope your wide receivers are. You hope they're high affinity. Every time the ball is thrown to them, they catch it and they score. Okay? Or if you like baseball, every time the ball is hit to them, they catch it and the batter's out. Okay? So high affinity transporters grab things really tightly and bring them into the cell with high efficiency. So I want to finish off talking about specifically bacteria and the two major groups of bacteria based on their cell wall. So most bacteria, right, we've already talked about one exception, most bacteria have a cell wall. Their cell wall is made of a compound called Peptidoglycan. It is a sugar amino acid compound. And we classify most bacteria into two groups based on a procedure called a gram stain. You will do gram stains in the lab. Gram stains take advantage of the different properties of the cell wall. There are a handful of organisms that you cannot gram stain. One, mycoplasma, which don't have a cell wall. Can't gram stain them. Some of the others include the mycobacterium, which have these really weird waxy layers. The stains don't penetrate properly. But most bacteria, you can call them either gram positive or gram negative. And they have different cell wall structures. So you do the same procedure to the cells, and you get a different result depending on the organization of the cell wall. So gram-negative cells actually have two cell membranes. So they have an inner membrane. We sometimes call it the plasma membrane. So it's an inner membrane or plasma membrane. Outside of that inner membrane, they have a very thin cell wall, a very thin layer of peptidoglycan. And outside of that layer of peptidoglycan, they have another membrane. We can call it the outer membrane. We can call it the cell envelope, too. And in that outer membrane, it looks a little bit different from a classic cell membrane because it has the addition of a compound called LPS. We'll talk a lot more about gram-positive and gram-negative cells, but LPS is a compound lipopolysaccharide. It also serves as a toxin. So it's why it can sometimes be more dangerous to have a gram-negative infection than a gram-positive infection. Because the cell envelope itself is toxic. Okay. A gram-positive cell has one membrane and a thick layer of peptidoglycan on the outside of that membrane. So gram-negative is like a sandwich. Two membranes with a little bit of peptidoglycan in the middle. Gram-positive, single membrane, thick layer of peptidoglycan. And we can see that in this cartoon. So on your left is your gram-positive cell. You have your single membrane, you have your really thick layer of peptidoglycan. When you do a gram stain in the lab, as long as you've done it properly, a gram-positive cell will appear purple under the microscope. There's a step where you add a purple dye, there's a step where you rinse, and there's a step where you add a pink dye. Because these gram-positive cells have such a thick layer of peptidoglycan, they hold on to that purple dye, and even when you rinse, they don't let it go. So they're holding on to this purple, you rinse, they're still holding on to the purple, you add the pink, and they're like, no, we're still purple. So they look purple. So here you see the coccus-shaped bacterium that are purple, gram-positive. Staphylococcus, streptococcus, bacillus. These are gram-positive genera of bacteria. Your gram-negative, you have your inner membrane, you have your little thin layer of peptidoglycan, you have your outer membrane with all of that LPS. They look pink. Because they have that very thin layer of peptidoglycan, as long as you do the stain properly, you add the purple, they hold on to it. You rinse, they let go. You add the pink, they hold on to the pink. It's tricky, it's all about timing. The first couple times you do a gram stain, they're all either going to be positive or they're all either going to be negative. You either didn't rinse enough or you rinsed too much. That's why we let you practice. You get the hang of it. And eventually you can get nice gram stains like this, where you can see here are some rod-shaped pink cells. So things like E. coli, Salmonella, Shigella, some of your gram-negative genera. So who cares? What does it matter? They have different cell walls. Ooh. I can occupy you for a couple weeks in lab, pretending you can care about the cell wall. Right? Here's your pink dye, here's your purple dye. It's super medically relevant. Again, cell wall inhibitors, they work really well against gram-positive cells because they have one membrane and they have this giant cell wall. If you give them drugs that say no more cell wall, the cell falls apart because of the osmotic pressure. But for many gram-negative cells, they have this tiny little cell wall. They have these two membranes. And you say, now you can't make a cell wall anymore. They say, that's fine, we got two membranes, we're still good. So many gram-negative cells are also resistant to cell wall inhibitors. So if you're a physician, your patient has this nasty infection, you're giving them penicillin, why aren't they getting better? Did you check what kind of infection they have? That could be why. Look for a different drug. The other implication I've already mentioned, that LPS found in gram-negative cells is toxic. What does it do? As a toxin, It turns on your immune system and causes what we call septic shock. It turns on all of your cytokines. It causes vascular leakage, leading to extremely low blood pressure, which can kill your patient. So it's really critically important that you understand the differences between positive and negative. I'm not just trying to keep you occupied for a couple weeks in lab. So there are reasons that we do certain types of experiments. And any of you who work in a clinical setting at any point in your careers will need to understand these differences. Okay, so long weekend. Don't come in on Monday. I won't be here. See you all on Wednesday.